So my name is Professor Paul Whitford. I'm in the physics department at Northeastern University. And what we work on is we study the dynamics of biological systems. So what we would like to understand is what are the physical principles that govern the dynamics of cells. To do this, we look at individual molecules and try to study how they go between different conformations or different shapes. And these shapes are important because some of them will be active, some of them will be inactive, so the balance between them and how quickly the molecules go back and forth ultimately governs what a cell does. And on a daily basis, what we do is we apply molecular simulations to ask these questions. So we propose concepts for how to think about these systems, then we formulate that in terms of a model that we can put into a computer, and then we ask the computer, what would happen if this were true? And at the end, we can take those results and look at what we know experimentally and say, well, are those basic concepts correct, or are we missing something about how the system works? And then ideally, we get enough of the story right where we can start to extrapolate and say what else is happening that we haven't seen already. So if you think about your body and you look at your hands, you could put your hand under a microscope and you would see cells. If you then zoomed in even further, you would see smaller and smaller components. You would see bacteria, you would see all sorts of little organisms moving around, cells doing all sorts of things. And if you zoom in even further, you see individual molecules. And that's what we are trying to study, is how do these individual molecules move, which then allow the cells to move, which then allow organisms such as yourself to carry out all sorts of fantastic functions. So if you look at this image right now, this is a picture of a ribosome. And each tube represents another molecule. And each tube actually represents tens of thousands of atoms. So together, this is what we would consider to be a molecular engine. Now, if you just take the structure, you could say you know where all the pieces are. However, if you look at a theoretical model with simulations, then you start to see that everything's actually moving. And this is what we are trying to understand, is how do all of these components move together? In this example, you see this big blue region that is moving from the left to the right. It looks somewhat subtle, but we'll zoom in again in a second and you'll see it in more detail. And what we'd like to understand is how does this big blue region move relative to the orange region, relative to the yellow and red regions as well. So in this example, what you will actually see is there will be very close interactions between the blue region and the red region. And what we have found is those specific interactions lead to this dramatic conformational motion that I was describing earlier. Now you can see it circled. And because of those interactions, this big blue region has to move out of the way to let the yellow and red tRNA molecules pass by. So one reason why there is a lot of interest in the ribosome is because every organism, bacteria, humans, you name it, they all have their own ribosomes. And one of the therapeutic applications is to figure out how to stop bacterial ribosomes while not stopping human ribosomes. And this takes the form of antibiotics. So if we can understand how this machine works at a very fine level, we can make antibiotics that precisely target bacteria without having any side effects for people.